Without hesitation, I'm Dan Biting. I'm an assistant professor at the University of Pennsylvania, and I lead the Microbiome database group. You're also going to hear, in addition to hearing from me today about the site, you're going to hear from Wojtek Besant, who's a software developer really leading a lot of the feature development and data loading for MicrobiomeDB. And as Omar has already alluded to, we are really a spinoff and leverage the infrastructure and really excellent work that's been spearheaded by the ViewPathDB group that develops resources for vectors and eukaryotic pathogens. So we'll talk today about this point and click interface for interrogating microbiome experiments. Okay, so what will we cover today? We'll start off with some site basics. Basically, where do you find out frequently asked questions? Where can you access or create your own free account, and why would you need an account? We'll also talk about how to just find and understand studies that you've found so that you can really appreciate what we have on the site and what we don't currently have. We'll also then talk about types of queries or searches that you can carry out on MicrobiomeDB. We'll then move into actually querying and visualizing the data on the website. We'll start off with some simple queries, and then we'll expand from those into more complex interrogations of the database that leverage not just one study or publication, but multiple data sets to ask more complex questions. And then we'll also take some time to query the data, not by metadata, and all this will become more clear in a few minutes, but by the relative abundance of bacteria. So we'll ask for samples in the database that have really high levels of, let's say, Staphylococcus aureus, or any of your favorite bacteria. And then at the end of the webinar, I will turn the steering wheel over to Wojtek Besant, and he will take some time to show you how you can upload your own data in the form of a biome file to analyze it privately in your own workspace. So we have two aspects of the site then. We have publicly available data we've already loaded onto the site for you to interrogate, and you can now load your own data. And then just to highlight that when we do these queries, we'll be doing them in a way that illustrates maybe some key kind of use cases that you might come to the database with. And these are driven by biologically motivated questions, right? How do we compare microbiomes across different body sites? How do we examine the microbiome trajectory during early childhood development? How can we look at the impact of geography or country of origin on how the microbiome changes during early childhood? And then can we identify samples with high abundance of one specific taxon? All right, so we'll do these live demo use cases to walk through and illustrate the main features of the site. And of course, if at any point in any of these queries you have any questions, just put them in that question tab. There's going to be lots of times where the website will take a minute or two to respond to what we're doing, and that's a great time to just kind of survey the questions and have a discussion. Okay, so, whoops, sorry. Okay, so before we actually go to the website, I want to highlight with the sort of landing page picture up here, I want to highlight where we're going to be navigating to and why. The first thing I'll point out is all the aspects of the site, virtually all of them, are accessible without the need to have an account. That said, having a user account does give you some flexibility and features that you can't get without an account. Most notably, it allows you to save and share your searches, which is really fantastic. And it also, now that we have our own private workspace set up, allows you to access your own workspace and upload your own data. So having an account is free, setting it up is really easy, and if you do set up an account, it works across all the ViewPathDB sites, which means if you already have an account on PlasmoDB or Vectorbase or other sites, those account credentials will already work. You already have access to MicrobiomeDB. We also have video tutorials set up, and we'll be updating those moving forward as well, but they give a quick 5-minute walkthroughs of different aspects of the site. The About menu at the top of the page has general information about the site, particularly how the site was designed, if you're interested in that. But I'll point you to, and we'll review these in just a minute, frequently asked questions that I think we get asked a lot. So we have answers to those FAQs under that About menu. We're going to talk at length about querying data sets, but I just want to point out a sort of basic pillar of how the site works, which is each data set or each study is represented by a card. And I've highlighted one of those cards here for a study of the microbiome, a longitudinal study of the microbiome in children in the Dhaka region of Bangladesh. Accessing any of these cards lets you query just that data set, all right? But we also have a card anchored in the first position of the website that lets you interrogate two or more or all of the data sets that we have on the website, okay? So that's an opportunity to do a cross-study query by interrogating this dedicated cross-study analysis card. Okay, we're going to go to the website in just a minute. Just a couple other sort of basic things. One is a little bit more detail about the cards. So all data is accessible via these cards, and the cards are really intended to be a synopsis of the study and what it is we're trying to do. The first is the study title and a link to additional, more lengthy descriptions of that study, papers that were generated from that study, links to the investigators that generated that data, et cetera. We have a bullet pointed description of the key aspects of the study, what it was and why it was conducted, how many samples were collected, and sort of the study design and where it was carried out. If there was a publication associated with the study, we have a link to that publication, and we have a tag indicating the type of study, in this case, H, standing for human. So I'm going to head over to the actual database website now. That's microbiomdb.org. And you're looking at really exactly what I just showed you on the slides. Here are the cards, and you can see that we can scroll across, and we have quite a number of studies represented on the site, most of them published, but some that are unpublished right now. And the first thing I want to show you is how we can find studies on the site, okay? So one thing you could do, of course, is scroll across and read a little bit about each of these, and I encourage you to kind of explore the site on your own. But we can also search for studies either by their category or by key terms. Searching by category searches essentially what's represented in the tag in the upper right, whether these are mixed studies that might have animal, human, and environment, whether they're rodent studies that were carried out in lab animal models, veterinary, for V stands for veterinary here, so large animal models of disease, and then human. And we have a little bit of data from an insect study as well. So those tags are all represented here, and clicking on any of them, like veterinary, will filter those data set or study cards down to just the ones that fit those criteria. So it's an easy way to sort of, at a high level, find the kinds of studies you might be interested in. The majority of the data on our site is human. And even once you've chosen a category, you can search within that category using free text. So I might be interested in diarrheal studies, and I just start to type diarrhea, and it immediately reduces my available study cards to 3 of them, all 3 of which are enteric disease microbiome studies in which many or some of the participants were examined because they had diarrhea. These free text search box searches the front of the card, but also searches these extended sample details, study details, sorry, that are available here. So clicking on that takes me to a more lengthy study details page that links to associated publications, the contact institution, and contact lab. In this case, Jeff Gordon's lab at we can also then search for kinds of studies like longitudinal. We can see that there's a lot of longitudinal data, even for human, represented on the site. We'll actually do a query of longitudinal microbiome data in just a little bit. Okay, so
So Dan,
heading,
sorry to interrupt. Just quickly, a few people had noted that they're having audio issues, but we did check with some attendees that we know on the call, and they do not have audio issues. So we're suspecting that there's either a regional issue with GoToWebinar or potentially specifically to that system and interacting with GoToWebinar. Just rest assured that we are recording this webinar, and you will get a copy of the webinar as a YouTube video. So if the audio does not get sorted out, that's unfortunate, but you'll be able to hear everything again as a video. All right, sorry, Dan, go ahead.
no, I'm very sorry to hear that, but happy that everybody will be able to listen to this one way or the other. Okay, so I'm heading back to the presentation just to highlight the key part of searching on MicrobiomeDB, which is you can search any data set or all data sets using our cross-study query by one of two ways. You can search by sample metadata, and we call this sample details. So these are details about the sample. Is it a stool sample collected from a child of age 5 with diarrheal disease? Those are all details that describe the sample or the individual from which that sample came. So that's one way to query our database and filter our database from thousands of samples to just the samples you're looking for. And you can just ask for those samples based on sample details. A second way to query in a very different way is maybe you don't care about whether it's a stool sample or whether it's from a dog or a mouse or a human, but you just want to find any samples that have the organism you're interested in at some reasonably high level of relative abundance, because you want to understand this organism in more detail, understand where you can find it. That would be a query by taxon abundance, and that's the other icon at the bottom of each card that looks like a little petri dish with bacteria growing on it. So sample details or query by taxon. Okay, so what I thought we'd do now is go ahead and actually use the site, navigate around, and in doing so, you'll get a sense for the key features and how it works. We'll get a sense for what we can and can't do on the website. So I'm going to walk through 4 use cases before I turn this over to Wojtek for his direct data upload. And they're all motivated by questions I think are interesting or important in thinking about microbiomes. So the first one is, how do microbial communities or microbiomes differ across different body sites in the human body? And so if that's our question, we have a data set on MicrobiomeDB that's perfect for addressing that. It's the Human Microbiome Project, or HMP. Many of you may be familiar with this. It's data from their phase 1 of the HMP. And we have data from V3, V5 sequencing. So this is just an opportunity for me to highlight that all the data we have on the website right now is 16S rRNA gene marker sequencing data. So we do not have shotgun metagenomics data, but that's something that we're actively working on right now. So if you're not familiar with this study, it was published initially in Nature, and it profiled over 5,000 samples from 242 adults, roughly half males and half females, across multiple body sites. And so this is an opportunity to ask, how well, using a web interface, can we rapidly query large consortia project data sets to gain biological insight? Okay, so we're going to head back over to the database website. I'm going to clear my filters here, and I'm going to search for HMP, and I can see the HMP phase 1 data from V1, V3, and V3, V5. And this is the data that has, in this case, 5,826 samples worth of microbiome data represented from 242 healthy volunteers. So I'm going to query that study by sample details by clicking on the little magnifying glass over the sample tube label. When I do that, I'm taken to the next sort of layer of the website, which is a filter page. And this filter page has a tree of all the sample details associated with these 5,000 plus samples, and lets me search that tree, click on different sample details or variables, and then filter the database from 5,000 samples down to however few I want, all right? So for example, I could search for sample type. And I can see there's a variable, a sample detail called sample type, and I can see there's mucus, saliva. I can see exactly how many of these 5,462 samples are saliva, and that's represented here as a sort of bar plot as well. And I could click on and choose certain subsets. Sorry, is there a question?
Yeah, I'm sorry to interrupt, but there's a request for you to zoom in a little bit to increase the size of your
Sure.
Okay.
Is that better? I'm gonna assume that that will be better. So
Okay, great, thanks.
Yeah, thank you. Okay, so in clicking on any of these variables, you can filter to any filter based on any of the values for those variables. But I'm going to do something really simple. I'm not going to filter at all. I want to analyze all 5,400 samples. So I'm not going to click on anything here. I'm just going to move forward yet again in the website. I'm going to click Get Answer here, and that'll take me beyond the filter page to the results page. Now, the results page has my samples that returned from my search, which in this case were all the samples from that HMP phase 1 V3, V5 data set. Each of those 5,462 samples are listed individually here. That's not so useful to me. I don't want to click through 5,000 samples, but clicking on any one of these shows me detailed information about that sample in an encyclopedic sort of way, what study it came from, all the sample details we know about that sample, and the actual taxonomic breakdown of the sample. What are the organisms detected? I could sort that right here directly and see that the majority of this particular sample, 30% of the sample, is comprised of Streptococcus. And so you can look at individual samples at that kind of level, but the real, I think, utility of the database is that we can take all of these samples that were returned from our search and all their associated sample details and pass them forward to data visualization apps, all right? To do that, we click on the Analyze Results tab. And what you're looking at when you go to the Analyze Results tab is a suite of data visualization apps. These apps, if you're interested, were all built in R, Shiny, and they launch directly in the browser. And launching any of these apps, what's happening under the hood is all of that microbiome data for these 5,000 plus samples with all the sample details that annotate those samples. All that information is being passed to those apps to let you, to give you an interface to visualize and query it. So I'm going to go ahead and click the Relative Abundance app. That's going to launch in the browser. Depending on how much data you're passing to these apps and the particular app that you're working with, there may be quite a lag while these apps load. Remember, in this case, it's got to consume over 5,000 samples. So we're going to have just a moment here where this app is fully loading. You can see it's telling us it's loading here, and it'll display in just a moment. In the meantime, this is a good chance to just check and see if there are any questions. Omar, I don't know if you've seen any questions pop up that would be good to answer now.
Nothing specific. Maybe while that's happening, we did notice that a number of people had solutions to the audio issues. One is that you just had to make sure you had the right audio output device selected, including in GoToWebinar, make sure that the audio output is properly selected. And another user indicated that that switching browsers from Firefox to Chrome worked, and so that's an option as well. So hopefully that will solve. And it looks like a lot of people who had audio issues were able to solve them by now.
That's great. Okay, perfect timing because the app now launched and we have data visualized. I'm going to explain what you're looking at, and then we're going to actually modify this visualization using the app. So what we're seeing here are the top 10 taxa from those 5,000 plus samples by median relative abundance. Relative abundance is here, so 100% at the top, 0% relative abundance at the bottom. And so we can see the top 10 taxa. That information alone is not so useful, and this is shown at the phylum level. So one of the things we can do with all the apps is change the taxonomic level at which we're viewing the data. So I'm going to scroll from high level phylum view down to genus. And although species level data can be viewed here, there's of course, concerns or questions about how accurate species level information will be from 16S marker gene data. Okay, so we can now see that Streptococcus makes up the majority by median relative abundance of the samples, and we can see those top 10 taxa still now at the genus level. Still not that informative because we haven't actually taken advantage of those sample details. That's here. So now I can see the value of what we've done, which is we passed not just the data to the app, but the sample details, the information about the samples. So now I can ask, well, how would this top 10 taxa look if sort of faceted or layered over based on body habitat? So I'm going to choose body habitat, and it's going to replot the data, this time showing me the top 10 taxa and how they differ across those 5 habitats. Now we see something really interesting, which is the ecosystem that's present at these different sites, colon, oral cavity, saliva, skin, and the vaginal mucosa, are very, very different in terms of microbial composition. Okay, we can see that, for example, the vaginal mucosa is dominated by lactobacillus, something that's been well described in humans. We can see that the colon is predominantly Bacteroides, that the oral cavity has predominantly strep and some fusobacteria, Prevotella, Haemophilus. And we can see that the skin is dominated by Cutibacterium, which is formerly Propionibacterium acnes, P. acnes, and also a lot of staph, right? So that makes sense. And saliva has a profile that closely resembles the oral cavity, which also makes sense. So very quickly, we're able to kind of make some pretty interesting observations about these different body sites with respect to the composition of the microbiome. Okay, so that's just one example of what we can do. I'm gonna head back here now, and we're gonna start to expand on this, and I'm gonna move a little more quickly because we're gonna be repeating some of the things I just showed you. And what I'd like to do now is take advantage of some longitudinal data on the site and ask how the gut microbiome really matures during early childhood development. So we're gonna take advantage of a data set that was published recently from Jeff Gordon's lab, looking at a group of individuals in which the microbiome was tracked longitudinally over the first 5 years of life. And they identified bacteria. Here's 1 to 60 months, so 5 years. Here are the bacteria that change as the children develop. So we're gonna head over to that data set. I'm gonna click on the header to go back to the home page, clear my filter, and we're gonna interrogate this Bangladesh 5-year data set. Again, a lot of data, 2,400 samples. I'm gonna query by sample details. I'm not gonna pick any filter parameters. I'm gonna hit get answer, and we're right back to where we were a moment ago, where we can analyze results, all right? And I'm going to actually, in this case, launch the Alpha Diversity app. Alpha Diversity is the diversity of the microbiome in a single sample, whereas Beta Diversity is the difference in diversity or composition between samples. So I'm gonna launch Alpha Diversity, and we're gonna ask how the diversity of the microbiome in these children changes from birth to age 5. Now this app takes a little bit of time to load as well. We don't see the visualization type displayed yet, so it hasn't quite finished loading. It'll tell us in a minute when it's ready for us to actually enter the axes, what we want to plot on the axes. So we'll take just a second. Any questions you could come off of mute to ask, you can put them in the question box as well. Okay, so we're ready to go. Okay. Yep.
So there are a couple of questions if you want to hear them now, or do you want to wait a little bit?
Let's wait 1 more minute and we'll get a chance to do that.
Sounds good.
So it's asking us to choose a sample detail for the x-axis. So what is it we'd like to plot the alpha diversity against? It knows we want alpha diversity, and it actually knows we want Shannon alpha diversity. We could pick different metrics, Simpson, Chow 1, or ACE. We're going to leave it on Shannon, leave it as a dot plot, and we're going to just tell it what we want to plot that alpha diversity against. So what I'm going to choose here is age at sample collection, and this is really, this might seem like a bad idea since there are 494 values for that sample detail. But what's going to happen with continuous variables like age is the app will actually bin the continuous variable into equal sized bins and use that to display the data more quickly. So I'm going to choose that sample detail, and we can immediately see that if we look at alpha diversity plotted on the y-axis against age, as these children develop and grow, so too does their microbiome develop and grow. Alpha diversity is increasing, eventually plateauing around 40 months, so just after 3 years of age. So that's pretty remarkable. It's a really nice view of microbiome development in the first 5 years of life. Maybe that's just, maybe that's just Shannon diversity. So one thing we can do is ask whether that looks any different if we change to Simpson diversity. And by the way, if any of you are wondering whether there's features or metrics available that you like and you check and find they're not, that's a great opportunity to contact us through the contact link, and we can see whether that makes sense to add that to the site. So here we see something similar but slightly different by Shannon, that we have an increase in diversity that plateaus again close to 40 months and doesn't really advance in diversity beyond that. Okay. So that's really cool, and we could also open up additional apps, each app staying open as its own tab. And so you could imagine having a sort of gallery of apps open that you could toggle through these visualizations. And if you're logged in, all of these searches could be named to give them a unique name. Once they're named, they can be saved and they can be shared. Okay, and sharing them actually just generates a link that you could send to your colleague and they would be able to see. So this makes it easy to have really transparent and reproducible searches on the website. Okay, so this is again a relative abundance of organisms. I'm going to toggle down to the genus level, and again, as I said, I'll move a little bit more quickly here, and that is taking just a moment to display. It's coming up now, and I'm going to look at age by sample collection, age at sample collection. So here is where we're going to see these bins really be useful. Okay, now what we can see is that it's binned the ages. We can see exactly what those bins are, and we can see that bifidobacteria, the most abundant by median relative abundance, really plummets as those children age in relative abundance. Whereas other organisms, Faecalibacterium, Prevotella, and Subdolingranulum, sorry, I can't say all these, come up with age, okay? These apps, by the way, are all, the actual graphics are all Plotly graphics, which means you can scroll, zoom in, you can double click to go back out, you can click off different aspects of the figure legend, so they're interactive plots. So this is really cool because what we've identified here are really age discriminatory taxa, and many of them align very, very nicely with what that were reported in this paper, which used much more complicated statistical methods to find these. Really important development of statistical methods in that paper, but nonetheless, simple web-based queries can give similar inferences. Okay, I think this is a good chance to take a couple questions, and then I'll show you how to do a cross-study query, and then I'm going to turn over the steering wheel to Wojtek. And,
all right, so
yeah, go ahead, Omar.
Okay, so there was one question, can we search by bacteria name? So I'm guessing
you can. Yep, and I'll demonstrate that in just a moment.
Okay, great. And the next question was, do you assign taxonomy, or do you take the taxonomy provided by the data owners from their own processing?
No, so we process, and this is under the frequently asked questions, we process all the data on the site through the same pipeline, and we use a Silva reference. So that's where we're getting our information about identity of organisms from the 16S data.
And the final question, I think we would probably save for Wojtek because it has to do with analyzing own data. So you can carry on, Dan.
Okay, great. So we're going to do a cross-study query now of multiple data sets. And the reason we can really do this is because of what I just highlighted in response to that question, that all the data is loaded uniformly through the same pipeline, and all the sample details are mapped to the same data dictionary. So we have age at sample collection in months for every study loaded, even if the investigators didn't initially record age in that way. Now, there's still all kinds of reasons, statistical reasons, study design reasons, why querying across studies for microbiome data without some sort of normalization, statistical normalization, might be a bad idea. But I'll highlight now one way we could carry out these cross-study queries and see what we can come up with. So I'm going to head back to the landing page, and I'm going to grab this cross-study query card, and I'm going to search by sample details. We see a slightly different way of filtering samples, and I'm actually going to filter now, whereas I haven't with the previous ones, because I want to choose certain data sets. I want to choose data sets that I know are longitudinal data sets, because the question that we're trying to answer here is, how does the maturation that we just observed in the microbiome, how does it really differ between countries? Okay, so we're going to try that by choosing longitudinal data sets that came from different sites. I'm picking these. I have 5 different studies here, around 8,000 samples, around 9,000 samples. Now, that's a lot of studies and a lot of samples. So one of the first things I want to check is that I'm, working with the same sample type. I don't want to compare stool to vaginal swab. So I'm going to choose just the stool samples here.
And Dan, how did you know these were all longitudinal, or did you have to go back to the
Yeah, you will find they're all longitudinal just by that data set search on the landing page. If you search
Perfect.
longitudinal, it will return these, and longitudinal and human. Thanks for asking that. And now I'm going to see, because I don't know what age ranges they studied in these different studies, so I want to look at age. So we can view continuous variables in this filter parameter here, and I can see the periodicity of sampling here across these different studies. And I can see that most of the sampling happened up to 2 years of age and then dropped off afterwards. It looked like some of the studies continued out to 3 years and less so out to 5 years. So I'm just going to either drag and select a range or enter the values here, 0 to 24 months. Maybe go 24.5 just to make sure we capture all those samples that were collected at 24 months. And now I've added these different filters, sample type and age at sample collection. I can toggle between them. I can clear them if I don't decide I don't want them, but it has dropped or reduced my initial 8,800 samples to 6,000, all right? So I feel like that's a pretty good place to start. I'm gonna go ahead and view those, and we're going to jump right into our visualization apps, and in particular that alpha diversity app, where we can look at the development of the microbiome over time. Okay, so while this is loading, I don't know if there are
There's
other...
one more question. Yep.
Okay.
So there's a question about whether you have pipelines also for eukaryota, so 18S or ITS sequences.
We do not right now. That's a great question. There's nothing to really prevent us from loading that kind of data. The methods we use for handling the 16S data should work fine for 18S or ITS, but we have not loaded any of those data types yet. Okay, so while this is loading, I'll just highlight that many of the data sets I actually am querying here, these longitudinal early life microbiome data sets from around the world, also have extensive clinical and epidemiological data available in one of the partner sites, also part of FewPathDB, called ClinEpiDB. So if you're interested in clinical and epidemiological data, and less so in microbiome data, you could head over to ClinEpiDB. We are working on ways to connect the two sites so that you could draw from information on one site while being at the other. Okay, so this is ready for us. I'm going to go ahead and enter age. Now we have a lot of sample details here, right? So I'm going to search for age at sample collection. And we're going to let this display. And then we're going to take advantage of these additional facets to get more information in our plot space. So here's our alpha diversity increasing with age across these multiple studies now. And I know that these studies were carried out in different countries because I could read that on the study cards. So I'm going to go ahead and search for country, and I can see that there are 9 countries represented in these data sets. So when I click on this, it's going to now plot a line. It's going to represent each of these dots depending on which country it came from, and it's going to plot a line to fit those points for the data sets from each country. Okay, so that might take just a second. I think it's going to plot. Maybe. Let me just try again. Maybe not. We'll give it 1 second while we're going here. I'm not sure why
I guess
it's not.
one thing to point out to you, to everybody on the call, is that you're passing a lot of data basically from the database into these sort of visual graphs that you're seeing on the page, and it's not summarizing the points, it's actually loading all the points. Is that correct?
That's right, and it's calculating, in this case, alpha diversity for all these data, and then it's partitioning them based on what we've asked it to. So it seemed to respond when I chose child's diet. So we'll let it replot based on child's diet, and then we'll change it. Oh, there it goes to country. So there seems to be a big lag here, which happens from time to time. I can hear my computer heating up. So I'm going to try to go back to country now and see if it'll respond to that. Great, so it is coming up. Okay, so this is pretty cool, right? So we can see this development or this trajectory looks similar for many of the countries, but there are some that really stand out as being quite different, including Bangladesh, which never reaches that level of alpha diversity, including South Africa. And so we could start to ask questions now about why the trajectory for children sampled from those countries may look different. Could it be just something technical in the way the samples were prepared or collected or stored, or could it be something biological? We could use other apps in the suite to ask what taxa really distinguish children from those countries or at specific time points in those data sets. And so there's a lot we could do here. Obviously, these apps are going to function in a way that's dependent on how much data we're passing to them. So passing 8,000 or 6,000 samples to a beta diversity plot might not make a lot of sense. It may be hard to really interpret, and it may take that app 5-10 minutes to actually display that data. So in the last minute or two, I want to just highlight this idea of querying by taxon abundance. And because that's a question we were asked, and something I highlighted in the beginning, that you could query by any organism instead of by sample details. So I'm going to head back to my cross-study query, but I clicked on that Petri dish instead of the sample details. And so I could choose data sets again if I wanted, but what I want to highlight is if I click samples, I now see not my sample details, but actually taxa. And that is searchable as well. So I could choose Prevotella. And once that types here, okay, I can now, my keyword search returns things at family, genus, and species level. So I could just look at the genus representation of Prevotella across every sample. So almost 23,000 samples present in the database. Now, this visualization, I can improve this by changing my bin width to be 1%, so 0.01. And even changing this to log 10. So these are customizable. And now I could actually choose just the samples that had 60% Prevotella or greater, all right? And we don't have time to do it in this webinar, but you could filter in this way on multiple taxa and really find specific samples that had a lot of your particular bug of interest or bugs of interest. And perhaps in doing so, find where might be the best kind of sample to start looking for that organism or trying to culture that organism or trying to study that interaction of that organism with other taxa in the microbiome. Okay, so I think we're at a point now, and we started a little late, so we're running a little late, but I think we're at a point now where I can take a couple questions on aspects of the site, but I also want to make sure that I give Wojtek plenty of time to go through what he wants to go through. So we finished this fourth use case of how you can search the database, all data sets or just specific data sets by taxon abundance, to find just those samples that have lots of your bacterium of interest. So that leads us to our final example, which is what about analyzing your own data, right? We have over 22,000 samples on the database, but what there's a good chance we don't have the data set you're most interested in. So how would you analyze other data not represented on the site? And with that, I'll turn it over. Wojtek, I think I need to make you the presenter.
Yes, I will.
I'm gonna do that now. So I am making you the presenter. If you have any issues, and why Wojtek's switching over, do we have any questions?
There aren't any additional questions right now, but I think it's probably a good idea to move forward. Maybe just to point out, as you did earlier, if you have questions, even if you think about them later on, and you think, oh man, I wish I asked that question during the webinar, don't worry about it. Just click on the Contact Us link on MicrobiomeDB, and I'm sure Dan or Wojtek will get back to you very quickly with an answer.
Absolutely.
Okay. So I am sharing my screen, and this is going to be about how to upload your own data to the site. And to do that, you need to have an account. I actually created one this morning very quickly, so I'm already logged in. Webinar, that's me. And in order to do that, like there's a tab at the top ribbon called Workspace. So this is, we haven't shown it in details, but this is where you can save your searches and strategies. And since the last release, you can also see your own data on the site through this tab called My Datasets. So I'm going to go there. And the first time you're going to go there, like you are not going to have any datasets. And it redirects you to this new upload section. It looks like that. So we will fill this in just a moment. I just want to show you what else is there. Like this is not a very long form. It explains what file it needs. And the format we've chosen is this Biome format, which has been designed to capture as many kinds of biological data as possible. So the same kind of data we have, which is 16S, but there are also biome files with whole genome sequencing and so on. So if you are generating data and you have been processing it with like your tools, you will most likely use something like Chime to visualize them. Each of these tools will have a way to create a biome file. There's also a suite of tools that provided by this project that lets you reformat your data in such a way that it's visible on the site. So, if you're like trying to prepare one of these and want to reach out to us for advice, please do, because we're very interested in what kind of use cases people might have for this. So what's good to add to the biome file, apart from the taxonomic information, is sample details, because as we have seen before, a lot of the functionality of the site is enabled by that. If you don't, you will be able to view the taxa as before, but like they will not be grouped in any kind of way, so it's perhaps less good. So I have prepared a small file that I will upload. I will just fill this in. So if you have a lot of these, like it's kind of helpful to say what they are. So I am going to navigate through my file system and find it. This is it. Yeah, it's just a small file. Like it has a few rows and some sample details. And I have taken it actually from the Biome Format project. Like they have examples showing you what these files are. And I'm going to click Upload Dataset. It shows me what it's doing. I'm not sure how long it's going to take. Perhaps long, perhaps not very. Let's refresh the page. Oh, that was good. So we have, of course, tested it with much larger files, and it might not take a second. It might take maybe 5 or 10 minutes, but it's showing you what it's doing, so you can kind of wait for it. So once the file is uploaded, like you can in this all datasets tab, and let's see what's there. Okay, installed and ready. Like this description I had filled out earlier. You can download it back if you forgot what you put in there. And then there are the searches. And these are going to be the searches which we have just seen, except with this file I have now uploaded. It's also a very good way of viewing files, like because like these are some tools to help you group them. So what did I put in? Okay, so like this is also a way to view files. See what's in them. I'm going to go back just to show you the other search. So this is a search by tax and abundance, and this one shows you what species they were and so on. Okay. Of course, the data is not that very meaningful. This is an example. So let's get all the samples, and the way it's all going to work is just as before. So all the apps should work.
And while that's loading, I'll just mention that all of those features of saving a search and sharing it, that all works for your privately uploaded data as well. So you can always have that in your workspace, but you can also share a search or an analysis you carried out with a colleague.
Yeah, so whatever you are able to do with the searches on the site, you are now able to do here. So say view by any taxonomic level, and the sample details you provided, now you can look through them. Some might be less useful. For example, like I don't think this is going to be very informative. Yeah, not very good. This one much better. So like it's one of these things that you get what you put in. So because the site is quite flexible, you can do a lot. And we have actually just started discovering what else, apart from the original use case, you can still do. For example, using the strategy panel, there is, I'm just going to show this, like it's, we have this from the genomic sites, but you're actually able to use this to view both your data set and another data set together in the apps, just so that they're there for context. Okay. So yeah, so this is it.
Great. So there's a question maybe while you're wrapping up here, either to you or to Dan. This was a question earlier as to whether you can, obviously, can you upload your own data? And the answer is yes, if it's in a biome format. But also within that question, does it have to be now only bacterial, for example, or can it be, or can it be, for example, fungal data or eukaryotic data, other eukaryotic data?
So as long as it's in this format we support, it's going to work.
So that's really great, obviously. So,
yeah, I'm not sure what the taxonomic tree would look like if you provided not bacteria, but some other things there, because we try to show it like kingdom, phylum, species. But there are attempts to make it work, whatever you provided, and I expect it to be quite flexible.
So maybe to today's audience, sorry Dan, just quickly to today's audience, maybe a challenge to you is if you have a biome file from an 18S, for example, analyzed data set or ITS, try it out, see how it works, and then click on contact us and let Dan and Wojtek know how it ran and what kind of problems you may have run into. Sorry, Dan, go ahead.
No, I was just going to mention, and just kind of following up on that. So there's no reason why you couldn't use this direct upload to look at data from a shotgun metagenomic study. We don't currently load shotgun metagenomic data, but if you have processed it and have it in a biome file, then that taxonomic information would be presumably easily accessible as a direct upload, and then examining it through the apps. I was also just going to mention it can be somewhat tricky to get your sample details into these biome files, but if any of you are familiar with how to use QIIME or Mother, these sort of standard pipelines for processing raw sequence data from 16S studies, they have features within those pipelines that let you sort of fold in your sample details into the biome file. And we have a link to that on, I'm actually going to change. Wojtek, are you okay if I change back to presenter?
Yeah, please present. Thanks.
So this may answer one of the questions while you're loading. One additional question came in is, how does what we offer or what MicrobiomeDB offers differ from QIIME?
Yeah, right. So I mean, we actually use QIIME in our data processing pipeline that happens, before data actually appears on the site. So all of the data you're looking at has been through a QIIME pipeline, but what we offer is what you do after that, right? So we pick up with how you filter, how you combine multiple studies, all those studies having been processed the same way with metadata or sample details being mapped to the same data dictionary, the visualization. So we're a post-QIIME, post-Mother workflow. And so under the FAQs, if you look at, how do I privately analyze my own data, that will give you an indication of how to do this. And we even link to our QIIME 2 protocol for how to make a biome file that has sample details in it. Again, if you have any questions, just definitely use that contact us link. There's nothing we like more than to get comments and questions from users. So I thought I'd just close with a couple highlights of where we'll go next with this. We do release new versions of the database every 2 months. That means new data sets. That means oftentimes new features. So be on the lookout. The site will develop and change and improve. Our current emphasis is really on clinically oriented data sets from maternal and child health. That's not to the exclusion of other data sets. We clearly have insect data. We have data from veterinary studies, but the majority of the data is currently from these human studies, many of which have data also available on ClinEpiDB for those of you who are interested in more extended clinical and epidemiological information. We enable tools, we plan to enable tools for loading and querying shotgun metagenomic data in the near future. That's something that Wojtek's working on. And we're always looking for ways to improve the filtering, the way you interact with filtering and visualization tools. It may be that we move away from Shiny apps at some point if we find visualization strategies and tools that are faster or more user-friendly. And with that, I think we are, at the end of the hour. I'd be happy to take any questions or hear your comments. And again, happy to also take those offline.
So yeah, there are a couple additional questions. So one is, would uploading our data to MicrobiomeDB be sufficient for a typical journal availability policy?
That's a great question. So if we head over to the site now, so I guess two things I would say. You should always make your raw data available, and that's often a requirement of publication. And we do not host raw data files so that users can come to our site and download them. In fact, if you click these download data links, you're not getting the raw FASTQ files, you're getting text file summaries of the microbiome composition from those samples. So you would still want to have your data on the sequence read archive or on ENA or one of these data repositories. We are not a data repository, but it is certainly very nice to have a data set presented on MicrobiomeDB ahead of publication. And so this PIH Uganda study is an example of a study where the data providers came to us and were interested in having the data up in time for their publication, which is about to come out. So that's certainly something we can accommodate, but I think you would still want to have the raw data available on a public data repository.
Great. It doesn't look like there are any other questions. Oh, actually, there's maybe one more. Okay, so there's a comment actually from David Roos, who's attending today. He just wanted to indicate that our release policy does not require release. The data provider is always, the one who makes the final call about whether they release their data or not. So
that's right. That's a great point. We never release data without prior approval from the data provider. And a lot of times when we work with data providers to load data, in fact, always, there's a close interaction with that data provider. We look over the way the data is represented on the site with them. All of this is done ahead of any release of the data to our live site. So yeah, absolutely. Thanks for bringing that up, David.
So there's a comment as well. Could a cross-study card be made to look at a lot of, I guess, looking at more different comparisons, I'm assuming?
Well, you can certainly customize this cross-study query in any way you want, right? You could choose data set, pick specific data sets that interest you, and then choose exactly which samples are going to be carried forward for that cross-study query. You don't have to choose all the samples. You can choose just the samples that fit the criteria you're most interested in.
Great. It looks like that's the last question we had so far. So I think unless you have anything else to say, Dan, we probably should end this webinar. And to note again that this has been recorded and you should get a link to the recording soon. We might edit out the beginning part, which was a lot of dead space as we fixed a couple of technical issues.
Yeah, thanks for hanging out through those glitches. I don't have anything else to say. I think Wojtek and I covered what we wanted to cover, but definitely reach out. Thank you again for tuning in, and I look forward to hearing from you.
Great, thank you, Dan and Wojtek. Yeah, thank you. Bye-bye, everybody.
Bye.